inoculum, the actual packaging. Okay, so we've got Alison Blackwell Tom Neat. We've got two different technologies or techniques to be able to do that. Nominally, this is supposed to be a workshop, so if you feel free to interact as we go along. Alison, Tom, I think, probably happy to answer questions as we go along or after they've done their presentation. So, just a very brief introduction then. So this is the problem we're addressing. Black leg that you're probably all very familiar with on the left, and even more familiar with the tube we've got on the right. This is just to illustrate some of the problems that we might come across with any kind of control solution, and that is that some of this infection is going to be internal. So this tube's been cut in half, and we actually haven't reduced the inoculum that's inside the tube. And this is infection that's occurring in the field or in the store where the infection is going in through the lens cells. We can talk about pathogen identification if it was necessary later on. If it wasn't quite right, we actually do have some pictures of messy potatoes. So here we have a bulk store, and underneath there is apparently a problem that they're trying to address. And this is the consequences of that delay. This is somewhat similar to Phil's.
I wasn't responsible for that.
No. I'm not saying where that came from. You actually flash it. I'll try and deal with it. So, we're dealing, as Phil said, we're actually dealing with that top bit of pathogen. At the moment, that's dealt with using integrated pest management. So, this book, which there will be copies of, which hopefully we can pick one up later on. And it's integrated. What it means is that we're looking to control the black bed disease across the entire agronomy storage cycle. Apart from that, there are some other methods that we could use to control the pathogen. You might see that chemical control is not mentioned on here. That's because, as far as I know, there's no good chemical method that we can use in storage at the moment. This bacterium would be very sensitive to penicillin, so if we were able to treat potatoes with antibiotics, job done. But clearly that's not very good. So we've got two methods: biocontrol and physical methods. Biocontrol in different ways of trying to do it, using other bacteria and other fungi. To be honest, it works in the lab, but not actually commercial scale, and that's our interest here. And then we can use physical methods to try. So many of you might remember trying hot water treatment. This bacteria doesn't like being heated up, and so that's quite an effective method. The problem is trying to heat lots of potatoes up, tons per hour, without actually having warm, wet potatoes, which is a fertile environment for the bacteria to grow. So without further ado, I'll get Alison to I have to say next.
So, yeah, thank you, Glyn. I'm from APS Biocontrol, based in Dundee, and we work on bacteriophage, which some of you may or may not have heard about. So what I want to do is introduce what bacteriophage or phage are. We haven't done much work in storage, but we've done some work at other points of the potato production cycle. So just want to kind of explain those briefly and then discuss some of the options available for storage. So has everyone heard of bacteriophage? Does anyone not know? Right, some nods, so that's right.
Alison, you know, there's a microphone there. There's the fans make a lot of noise, so you can set the fan off with it.
Yeah, here we go.
Technology failure.
Just hold on.
I'll shout. I'll shout. I kind of got the box. Okay, so bacteriophage. God, that's a bit loud. Does this work? Can't get the tools these days, can you? Anyone got any spare batteries for this?
Yeah, we'll have to sort that out later.
Okay.
Just say next.
So, bacteriophage are naturally occurring viruses. They're everywhere in the world. So your glass of water you're drinking at the minute probably has 500 or 600 bacteriophage in them, and you're drinking and eating them the whole time. And to date, at least, no one has dropped dead from bacteriophage poisoning. So this could be the first case, hopefully not. Most abundant organisms in the world, they live in bacteria. They only infect bacteria, and every bacterial species has one or more bacteriophage associated with them. They don't do anything else any harm. They're very, very specific, so they only target bacteriophage. They've got a great history of acting as potential antimicrobials, primarily in the medical field. So the Russians first discovered bacteriophage in the 1900s, early 1900s. And today, if you've got an ear infection or, I don't know, a runny nose or something, you can go to a clinic in Russia and you can be prescribed a bacteriophage preparation instead of antibiotics. Europe is a bit further behind and a bit cautious about bacteriophage, but we're trying to educate them slowly. Completely harmless to, works now, completely harmless to humans. The good things, it's a very specific, almost down to the level of a strain of bacteria, good and bad for us. They're very safe, biodegradable, and they work very quickly. Their mode of action relies on the way in which they reproduce. And I've got a little model here. So this is our bacteriophage. It's a kind of spiky thing with legs. And all it is really is a protein coat with a bit of DNA inside of it. I'm not very good at drawing, but these blue blobs are bacteria, pretend. What happens is the bacteriophage attaches to the surface of a bacteria. It recognizes some receptors on the bacteria and injects its own DNA into the bacteria, and it takes over its production cycle. So the bacteria itself becomes a bacteriophage production factory. And as soon as you've got lots of bacteriophage in it, it kills the bacteria by bursting, and as you can see, they then go on to infect other bacteria. This cycle takes only hours, and you can clear a bacterial culture with phage in a matter of hours. So it works very, very quickly. So how do we find bacteriophage? We say they're everywhere, but they can be quite difficult to find. Start off by isolating the bacteria. So in this case, we take potatoes. We isolate on pectolytic plates. So these little red blobs are all Pectobacterium, responsible for soft rot. We then, can you press the next button, please? We then do something called enrichment, so we take environmental samples and we mix them with our bacteria we've isolated, and we look for bacteriophage, which we know are going to be there. This plate on the right-hand side is started off as a plate of bacteria, and then all the little clear areas are bacteriophage, the viruses which punch a hole in the bacteria. So we know that these clear areas or plaques represent bacterial killing. And we can take these, next please, we can take these bacteriophage and we can grow them up in the lab until we're ready to use them in the factory or the field. Next please. Advantages we've already looked at quickly. Bacteriophage are very safe in your food, in your water, everywhere. Very, very specific. They don't infect other organisms. They don't have any environmental impact. We can use them for both mainstream and organic agriculture. And the right mix of bacteriophage will continue to work, not like a chemical which has a one-hit effect and then oxidizes and goes away. Your bacteriophage will sit on your tuber or whatever your plant you're targeting and continue to work as long as the target bacteria are there. Next, please. So, options for using bacteriophage against spoilage pathogens in potatoes can be in the field, at planting, during the growth cycle, or maybe at harvesting. Next, please. Maybe during storage, which is the focus today, or perhaps in the factory. Next, please. And next. Back. Back. So the challenge for us has been to develop a bacteriophage technology which could be applicable across the potato cycle. But we started where perhaps it's easiest to do so, and that's at the packing stage. So we know that rots are the biggest cause of customer complaints that the supermarkets get about packed potatoes. And that costs the industry significantly in terms of fines, in terms of warehouse rejections, etc., etc. So what we've done is try to prepare a bacteriophage product which can replace some of the chemical biocides the packing industry currently uses. The product is called BIOLYSE. Some of you may have heard of it if you're in the packing industry. It's a mixture of bacteriophage which is applied during packing and significantly reduces rot development post-packing, so reduces shelf life failures and also rejections at the customer end. So this is your washer barrel, this is your potatoes coming out of a washer barrel, and we have a spray bar which goes on the roller table just before the bags go into packing, and they get a fine mist of BIOLYSE, a bacteriophage product. These are examples of potatoes we might see. So you've got tubers coming straight out of the washer barrel, nice and mouldy. Those which may just have a water spray, again beginning to rot. And this is a BIOLYSE treated potato 10 days after treatment, and you see it's nice and dry still, no development of rot at all. Feedback from the industry has been very good. 2 examples here, some Estima phage treated here after 7 days, and this is a control. Some Desiree, again, treated potatoes, untreated potatoes. And this is some customer data where we encourage our customers to get involved with the whole assay process because it's a new technology and we need to build confidence in the industry. So all of our customers take tubers pre-phage treatment and post and measure shelf life rots. The purple bars are the untreated tubers, and this is a rot development during their shelf life studies. And the green bars are the phage-treated potatoes, the same batch of potatoes, and the equivalent rot development. And you can see we're significantly reducing rot development with the product. Next, can you just press the... Okay, that's fine. We've done a bit of work in the field as well. So what we want to do, obviously, is to stop the packer having all those problems with rotten potatoes. We want to go back into the field. These are some pilot results which we're continuing to develop within our Agritech Catalyst program at the minute. In this case, we took some Desiree seed, we bashed it around, we inoculated it with Pectobacterium, we then treated it with Bacteriophage, planted it, and this was the one of our trials with the degree of blackleg developing in the field. So we've got about 12% in our control untreated seed, and significant reductions with increasing concentrations of bacteriophage. So that's quite encouraging that we might be able to actually reduce rot development in the field. And as I say, this is ongoing as part of our TSB project at the minute. Finally, during storage, we've not done very much work during store, but I've got one example here. This was a company that came to us and said, we've got a whole batch of Lenormand seed at the minute and it's rotting in store. They gave us some to test. We had about a 70% coverage of the Pectobacterium we isolated from it. They graded it out, took some of the cleaner tubers, we treated it with Bacteriophage, and it was put back into the store. And it was very, very mucky seed to start with, but what we saw over time, and this is data from 6, 12, 18, 24 weeks, we had an increasing level of rot development in our control seed, and at least a reduction with our phage treatment. So in summary, we've got good potential with bacteriophage and natural controls for reducing the development of bacterial rots in potatoes. Huge variability of the pathogen is complicated, but so what we're doing is producing mixes of bacteriophage to cope with this. We've got a commercial product which is used in packing, and happy to talk to anyone about that today. But the key application of the technology, I think, is going to be in the field, but also potentially during storage. For that, I think we need to look at application method, timing, etc., and that's perhaps what we could discuss here. Glyn's telling me to stop. So, to you.
Thank you.
Thanks very much for that, Alison. If you've got any questions for Alison, just while I'm setting up his presentation, feel free to ask a question.
Yeah.
I may have misread it, but in the trial where you applied your BIOLYSE to the seed and blackleg in the field, you had a result for rots as well, and you said there was no difference in rots in the different treatments. So I just wondered
That was in the harvested tubers. No, there wasn't in that trial.
would you have expected there to be?
Would have thought so, yes. But I think the blackleg plants in the field were actually rogued out, so in some way the source of those potential rots is gone. So.
Okay. Tom, do you want to introduce yourself?
Yeah.
Tom?
Let's borrow this a minute. Swing this round here a minute. Oh, don't get lost too much. Hi, good morning. My name's Tom Neat from Techneat Engineering. In the past, we've worked on a lot of chemical application machinery, bits and pieces, and a bit like Michael did earlier, looking for future things, future directions for our business, where we should look to go. And one of the things we've invested in is product development and looking at different areas. And one of those areas is pulsed UV surface sterilisation or surface disinfectant. It's a project we've been working on sort of behind the scenes for trying to develop the technology for the last sort of 3 to 4 years. And in doing so, we required better expertise, and to gain that expertise, it seemed logical at the time to bring the specialists involved to form a separate company called Apollo UV. So Apollo UV is made up of myself and a number of other directors with specialities within the UV and the lamp technology and the electronics field. Should we go to the next one? Pulsed UV, what this is basically a waveform, a light waveform that's produced by this lamp, and I'll pass this round. you don't really want to take it out of the bag, but and be quite gentle with it. But it's worth quite a lot of money. But basically, the lamp that we have there runs at about 3,000 volts, and the reason why there's a little cage on the outside of it, we put a little trigger voltage to that of 20,000 volts, which causes the lamp to arc. And basically, in that arc, it's a bit like a welding torch. You're basically flashing it on and off 3 times a second, and in that, it's emitting this waveform that you see here. The UV part of the wave is basically from 300 nanometers down. It will come down here further. That's where we're getting up the sterilization effect. Everything you see to the right-hand side would be the visible light that you'd probably see as the bright flash. Next. The difference between pulsed UV and conventional UV is basically in our pulse of 3 milliseconds where we generate that flash, we generate a huge peak power hitting the surface of whatever we're hitting. And in that peak, it will totally change or destroy the DNA of the bacteria on the surface. And it's a well-known technology that's been out there. You will be aware within the food processing industry of UV tunnels. The advantage we have is that we don't have the exposure time. So basically within that 3 milliseconds, it's like a photographic flash. It's just bang, and that should give, as long as it hits the target, a surface sterilization or disinfectant. Next. Some early work that we've been working on, some dip slides, some crop safety, seed tuber or bacterial reduction, and shelf life extension. Now the work, we've spent a lot of time in developing the machinery side of things. We now need the industry, people like yourselves, to try things and to see what better results we can come up with. But if we click to the next one. So dip slides, we can see our control, our two pulses, 4 pulses, 6 pulse and 12 pulse. So basically all we're saying there is we're getting some level of control, good level of control. As a rule of thumb, you're looking at about 1.9 to 3.5 log reduction in a single flash, depending on the target that you're actually hitting. Next. Here we show some blackleg on Petri dishes. This work was done with Glyn. Basically, Glyn provided us with the Petri dish with the Pentobacter solution. And again, you can just see the difference between control back down to single dose. Second dose didn't give us much more reduction and additional doses. We also did a treatment back end of 2014 on some ware potatoes, where we put them through a roller table rig with an array of lamps on the top of the unit. And we got a significant, I think it's right to say, statistically significant difference between treated and untreated, both in the severity of the rot and the incidence. And this was in an environment where we tried to put them in a bad case scenario in a warm environment, in a damp environment, to try and bring on the rots. Next, please. As far as crop safety goes, we've grown several tons of seed, so both as well as store bacterial reduction. We also looked at seed. We're also worried about would we affect the germination, would we affect the survival, if you like, of the seed tuber. And in this instance, we basically did treatments where we were directing the flash directly into the open eye of the seed tuber, and it had no effect at all. Just a picture of a field growing. The one with the 2 canes next to it was a control. The one to the left is the treated run, but that's shelf life extension. Again, simple little test. It's not any great level of data, but basically left-hand side we have the control with about 50% rot levels, obviously stored under harsh conditions, and the treated side on the right-hand side to a single tuber that's rotted out in the small batch. Next. This is a seed trial that we ran last March, crikey, with our small roller table in the centre there with the Apollo UV on the side of it. Underneath that cover there's a light unit, and that shows the installation there. A summary. Now these results were done by the farmer's son. He was doing his undergraduate thesis up at Edinburgh, and he wished to use it as part of his thesis, so we've worked with him on that. The variety he was using was Desiree, 4 replicants. Basically, this is a summary of his results. Emergence, 100% fine. All plants look healthy, good leaf shape, no visual differences. Ground cover fine. The downside of last season, and it's a bit strange because the reports we get back in East Anglia is that there's issues with blackleg coming from Scotland. But we didn't see any blackleg in the treated or the untreated during the growing season in this instance. And we also had another 3 trials that are running where SASA were doing some field walks. They saw the same. In the treated and untreated last season, during the growing season, they didn't see the blackleg. Next. What he did see was, although the last field walk was done on the 26th of July, come the 2nd of September, he did see a difference. Now, probably not a significant difference in fairness, but there was a difference there, both in the instances of blackleg showing up on the black stems, and also in the major rots, 16:3. Next. What he also sort of mentioned as well and put in his results was the size distribution. And again, now it's only on this one test, but if it's of any value to you, it seemed to give more, in this particular trial, more mid-ranged tuber sizes. Just to give you a little bit of run through of the history of where we sort of have come from with the units. This was our Back to the Future sort of model. As I said to you before, we were running at 3,000 volts and 20,000 volt trigger voltages. And sort of as mechanical engineers, we somehow put this together. With the expertise of bringing other people on board, we've then further developed it to the cartridge system that you see, if you can see here, which is like a self-contained unit, can be put within other things, you know, could be put with another manufacturer's roller table or whatever. So you can put together. Next. And then here we have the seed machine on the right-hand side, which we took to Scotland. Slightly different. That hasn't got that inlet chute on it now, but that's it. And then what we have here, which we hope to be able to make available to potential growers or packers or whoever to go around, is a small batch machine where we have various things that we can trial with this. We can put a small roller table on there. We can do small batches, maybe 10, 15, 20, even probably 100 kilos of product through. But it's something that can go on a bench, quite easily plug in, put it in the back of the car and take it out to a packer or a grower to do that. Next. Future work. We've got a little ongoing trial with SASA regarding skin blemishes. We went to see them 18 months ago regarding blackleg, and they was putting more pressure on us to say, okay, what about skin blemishes? Now, the issue with the skin blemishes would be chasing fungal spores as opposed to bacteria. The data would show that we can treat fungal spores, but we need a more higher-powered lamp. So we're working on a higher-powered lamp. We're also developing a wear machine. One of the issues that we have, and one of the potential things is, the same with any application machinery, you've got to hit the target. And for us, the initial machine was developed around small seeds. When we went to wear crop, you were passing it through, and obviously it wasn't rolling the same way on the same roller table. If the crop doesn't roll, we won't treat it. It's important for us that the crop is rolled as it goes through underneath the lamps. Yeah. Oh, okay. If you go back one, I just. And then seed assessment, basically it's just having the industry to help us quantify what the bacterial reduction is. It's, again, as an engineering company, we need your support to help us to do that. Next. And obviously for UV, it's not solely for potatoes. There's a whole different range of things, and UV is used in packaging, water, eggs, salads, fruit. Some of those we've already worked on. Room disinfection is obviously an area of concern regarding hospitals and hotels and what have you, and equipment disinfecting. Any questions, please? Pardon? At the minute, we haven't got a machine where we can say there's a machine and there's a cost. All I would say, the running costs are very low. This light unit here will run off a 13 amp plug at 3 flashes a second. Obviously, to treat a wider roller table, we put the lamp units in an array across the roller table, but it's still not excessively. But the typical cost, I can't give you a cost in electrical usage. The lamps, we expect to get at least a year to 18 months worth of flashes out of a lamp, but it obviously depends on the usage. But each lamp is good for 3 to 4 million flashes. So.
What about soil coverage on?
Yeah, that's a big thing. Yeah, I mean, with UV, if you put an umbrella over the top of it, then you're not treating underneath the soil. So that's important to roll it. It's important for it not to be covered in soil, or if it is covered in soil, it will treat the surface of the soil and not the surface of the tuber. That's something that is a potential problem. It's no different as well, though, to some spray treatments for, the good thing with our process is that our droplet size, if you talk of it as a spray, is infinitely small. So we are getting perfect coverage. And again, Glyn's given an example before, was if your droplet size from a spray treatment, the bacteria's got to walk from here to probably back to Cambridge to actually pick it up. Whereas with the light treatment, it is on top of it, as long as it isn't covered in soil. Okay.
Okay, thanks very much, Tom and Alison. They're both around over lunchtime if you wish to catch up with them. I think you'll agree that's two really nice complementary techniques, possibly, for controlling the pathogen, at least post-washing, or and potentially in the field and into the store. If there's any other questions, perhaps you can catch up at lunchtime, which it now is. So thanks very much.